Hey guys, welcome to another video for biology. In this video, we're going to be taking a look at cell division, or the cell cycle that is. So these are the three main objectives that I'm going to cover in this video. First, what is the cell cycle and the purpose of it? The cell cycle broken down into its subdivisions. So you're going to see here that there's basically two subdivisions, two layers of information that we're going to look at in this video. And then the checkpoints, the quality control of the cell. So let's go ahead and dive into the cell cycle here. So it's helpful when talking about the cell cycle to picture a pie. The first slice of pie that you see there, we're going to call mitosis, the M phase. This is probably the most popular, the most well-known aspect of the cell cycle. We will make a whole video dedicated to that entire process in detail. And then what comprises about 90% of the cycle is interphase, the biggest chunk of the pie there, if you will. So as we look at these different aspects of the cell cycle, you can look at interphase and break it down into a subdivision of three component parts. The first being growth phase, you have synthesis, the S phase, and then you have G2, the growth phase, the second, if you will, okay, which is very much related to the first. All right, so let's take a look at the G1 growth phase. This lasts about 4 to 6 hours. And in this phase, you have organelles that are being copied, duplicated. So you have endoplasmic reticulum, you have lysosomes, mitochondria that are being copied over. Again, the goal of the cell cycle is to make a brand new exact copy of the previous parent cycle, cell that is. And it does it through the cell cycle. So the first part is copying all the component parts of that particular cell, with the exception of chromosomes. No DNA information is being copied at this phase. But when you get into the S phase, or the synthesis phase, now we're talking about copying DNA information, genetic information. And this is the cycle, or this is the phase of the cycle that lasts the longest. It lasts anywhere from 10 to 12 hours. And again, it's replicating DNA. So all those millions and millions of lines of code in the chromosomes is being copied very carefully. Okay, everything is being duplicated. And then finally, we have the G2, the growth phase number 2. And in this one lasts anywhere from 4 to 6 hours. And it's very much related to the G1 phase in that it's essentially the final prep for mitosis. So we are still copying some of the organelles, but for the most part, it's prepping everything for that final mitosis M phase. And then we get to the M phase mitosis. This phase, surprisingly enough, is actually the one that runs the shortest. It's less than an hour long. And in this phase, we are copying, duplicating DNA. There are subcategories. You can break this up into a subdivision, 4 subdivisions. So mitosis can be broken up into what will be called PMAT. Okay, PMAT being prophase, metaphase, anaphase, and then finally telophase. And again, we're going to make a video detailing each one of these components in mitosis, PMAT, in great detail. So for now, we're just kind of getting an overlook or an overview of the cell cycle. And again, the PMAT comprises, put them together and makes up mitosis, the M phase, that is a small portion of the whole cell cycle. Okay, so yeah, you have the subdivisions, and it lasts anywhere from 20 to 24 hours. Each cell, depending on the cell, of course, this is an average, the cell will divide, that cell division will last 20 to 24 hours. Okay. So all that comprising the cell cycle. Now, before we move on, as we finish up, I do want to take a look at one other component that is very important. That's the checkpoints. Okay, so these checkpoints are essentially points in the cell that have to do with quality control, making sure that all the organelles are properly copied, making sure that all the DNA is properly copied. I mean, that's a huge undertaking when you stop to think about it. And the cell has various checkpoints that are controlled by enzymes, so other proteins that are basically going around checking to make sure that everything is a go. And so, Some of the things that it checks for are there sufficient nutrients, adequate cell size, is there damage to the DNA? If there isn't, then you get the red light and the cell will go into what's called a G0 phase, G0 phase, and we'll talk about that in just a second. But if you get the green light, everything is a go, then the cell will continue from G1 to synthesis. And as you remember, it's 10 to 12 hours synthesis. And even during the copying of the genetic information in the S phase, the synthesis phase, there are checkpoints within that phase itself. There's a lot of quality control aspects to the cell being divided. And then we end up at the G2 checkpoint, where again, make sure that everything has been replicated properly and completely, that there's no damage. Because any damage that is done to a cell, when it enters the M phase, is going to get passed on to that new cell. So now instead of having one bad cell, you're going to end up having 2 bad cells. And then those 2 bad cells will make 4 bad cells, and then 16, 32, 64, so on and so forth. Okay? So when you have the green light, the final phase, the M phase, the division phase of the cell via mitosis goes through. Now, so if a cell does not properly have all the proper checkpoints, it does go into a G0, that G sub-0 phase. Now, most cells are actually in the G0 phase already anyways. For example, you have muscular tissue, or you have nervous tissue that spends its lifetime in the G0 phase, does not replicate, does not divide. The number of muscle cells that you have are the same ones you had when you were born. Okay, the question is whether that muscle either gains size or not. But even though it gains size, that does not mean that it increases in number. Same with your nervous tissue. So most of the cells spend their time in the G0 phase. Now there are other cells that are in the G0 phase, but depending on some external trigger, maybe it was like for example a liver might get damaged and might engage, its liver cells might engage now into a G1 phase, an S phase, G2 phase, depending on whether it needs to be healed or it needs to go and heal something else. So, or a skin cell that gets damaged. Okay, that cell might be in G0 phase, or maybe some cell dies off by apoptosis. So something that was at one point in G0 will then get triggered to go into G1, and then S and G2, and so on and so forth. So cells are constantly changing depending on the needs of the body. And again, there are some cells that don't, like nervous tissue or muscular tissue, that will remain in G0 phase forever. And so that's pretty much it for the cell cycle, the overview of the cell cycle. It is important that you remember the main components of the cell cycle and then the subdivisions of those cell cycles. And then we'll talk about each of those individual components in greater detail later on. All right, thanks, and we'll catch you in the next one.